The title of my presentation is Harnessing the Potential of Microalgae, a Step Towards Meeting the Climate Crisis in the 21st Century. Like Dr. Paul said, I'm Dr. Anirban Gupta, Assistant Professor at the Department of Biotechnology, School of Science and Technology, the Neotia University. Now, since the dawn of the Industrial Revolution, we have seen a period of unprecedented growth that has been fueled by technological metabolism and utilization of fossil fuels. So how does fossil fuel work? We burn fossil fuel. Burning produces heat. Heat converts water to steam. Steam moves turbines, and turbines produce electricity. And this has been the invisible dance since the Industrial Revolution that has powered civilizations. We have moved trains, planes, we have reached the moon. We have moved mountains and rivers. We have even changed the rotation of the earth by a slight degree. So that is the power that has been generated through utilization of fossil fuels. But this has come at a very heavy price. Burning of fossil fuels has also caused the accumulation of greenhouse gases, particularly carbon dioxide, in the atmosphere, which acts like a magnifying glass held on the entire planet that is causing the planet to heat up. Now, 2.5 decades ago, climate change seemed like a very distant problem. This was perhaps the most iconic image associated with climate change, that Arctic snow was melting, the polar ice caps were melting, and polar bears living thousands of miles away were losing their habitat. And we did not care much about them, honestly speaking. But with the progress of time, climate change has caught up. This year, we saw one of the most devastating wildfires in one of the richest cities in the world. Our own country was plagued by drought and other extreme weather events like cyclones and super cyclones. I remember in 2020, there was a devastating cyclone called Amphan, which ravaged my own community. For almost 75 hours, there was no power supply in my home. Many others faced a similar situation. Therefore, it is fair enough to say that climate change is literally at our doorstep, and we need to act, and we need to act very fast. Many may feel that climate change is perhaps a geopolitical issue. I beg to differ. It is not a geopolitical issue. It is an engineering problem. It's a technological problem. We need to come up with technologies, sustainable technologies, that can meet the challenges of climate change. So the crisis very much looms upon us, and we need to find viable technological solutions to this very real existential threat to human civilization itself. Now, inspiration and motivation, the global challenges. Recognizing the challenge of climate change, the very existential threat to humanity, nations of the world got together and signed the Paris Accord. And following up, in the United Nations General Assembly of 2015, the Sustainable Development Goals, 17 Sustainable Development Goals were identified that we need to address in the 21st century. Now, I always say in my class and in all my lectures that 21st century problems require 21st century solutions. So today I would like to focus on many of these sustainable development goals, particularly related to climate action, affordable clean energy, good health, clean water and sanitation, and perhaps life below water. So, like I said, that climate change is a technological problem that requires an engineering solution. Algae, particularly microalgae, are increasingly emerging as a viable, sustainable solution to that problem. The problem of climate change is caused by the accumulation or the overaccumulation of carbon dioxide in the atmosphere. And there is a very ancient biological process, that is photosynthesis, that can fix carbon dioxide from the atmosphere and convert it into biomass. Carbon dioxide is essentially inorganic carbon. So when we speak of microalgae, we basically say that yes, microalgae can perform the process of photosynthesis, by which inorganic carbon can be converted to biologically available carbon. But the scope of algal biotechnology goes beyond that. There are several vital ecological functions, biological functions that microalgae can perform. They can be an extremely viable source of biofuels. They have very high growth rate and photosynthetic efficiency. Microalgae can function as fish feed or other high value products. Microalgae cultivation is a comparatively rapid process and is a fairly easy process to operate. Microalgae can be a source of oxygen, and it can, please excuse me, fix and sequester carbon dioxide from the atmosphere. So let's look at the algae cultivation system, an integrated algae cultivation system, and what is the promise of microalgae. Now, carbon dioxide, like I said, is produced in power plants, the thermal power plants, and microalgae, interestingly, can be grown in the presence of wastewater. So we can combine the flue gas, that is the gas emissions produced from thermal power plants and wastewater to culture microalgae. We can do that in open systems or closed systems. And once the microalgae has been cultivated and cultured, we can harvest that microalgae. And we can either directly use it as a source of fertilizer, which would be a very easy solution. But then again, we can subject it to some form of pre-treatment and extract carbohydrates and proteins. We can subject those carbohydrates and proteins to fermentation to get bioethanol, methane, or hydrogen, all very clean fuels. Or we could directly go for oil extraction. And by various processes like transesterification, hydrothermal cracking, or any other treatment process, we can get biodiesel, jet fuels, gasoline, that is petrol, and other high energy compounds which can serve as biofuels. So this is the basic schematic where we see that flue gas and wastewater can be combined in microalgae cultivation systems, which will be followed by harvesting, followed by biofuel production. Now, this is a bit of a schematic which will help us to visualize the very concept of algal biorefinery and algal biotechnology. If we pay a bit of close attention, we can see that the flue gas that is coming out from the thermal power plants, they can be used in presence of sunlight by the various species of microalgae. Interestingly, microalgae is one organism that can be grown in presence of wastewater. So we don't necessarily need clean water or expensive culture media for large scale cultivation of microalgae. Now, this, like I said, large scale cultivation of microalgae can be done in two different systems. One is known as the photobioreactor, the other is known as the raised pond. This is an open system. This is much cheaper and easier to operate. It's less energy intensive. It is less labor intensive. However, it is extremely susceptible to contaminations. However, the closed systems, which are slightly more expensive, but the productivity is much higher. And of course, they are much less susceptible to any form of contaminations. We can directly subject this microalgae to fuels to make fuel cells. Now, I must confess that this is largely a theoretical concept, and we have only working prototypes in some of the laboratories across the world. This has not been applied on a very large scale to produce bioelectricity directly using microalgal fuel cells. However, what has been done and what has been accomplished and what continues to be explored on a fairly large scale across the world is using the algal biomass. What do we do with that algal biomass? We get the algal biomass, we harvest it, we dry it, we can extract pigments. Now, some of these pigments can be very high value compounds like, let's say, carotenoids, which have high antioxidant properties. Some of them are antimicrobial or anti-cancer properties. We can use the microalgal biomass for fish feed or poultry feed or biofertilizers. This can be one option. The other option can be extraction of these biopharmaceuticals. So once again, these are very high value compounds. And of course, biofuels. The advantage of using microalgal biofuel is that here we are getting the carbon dioxide that is being emitted from the thermal power plants or burning of fossil fuel, and we are directly converting back that carbon dioxide into some form of biofuel. Another option is use of biopolymers, which can function as alternates for conventional petrochemical plastics. So once again, that is another option that we have. So microalgal biomass, which forms the central character of any algal biotechnology, has a plethora of diverse applications ranging from electricity to fuel, from pigments to fertilizers. Now, I have enlisted some of the high value compounds ranging from vitamins, antioxidants, antimicrobials, antifungals, etc. So these are some of the high value compounds which can offset the cost of microalgae cultivation. Coming to the process of microalgae cultivation itself, like I said, there are two basic technological strategies. One is the closed system, the other is the open system. Here we have some examples of closed system, starting with a very simple bubble column reactor. It's a vertical cylindrical bioreactor. At the bottom, we have some kind of air spurger, which will spurt sterile air into the culture medium of the microalgae. Now, this can be slightly modified into draft tube uh, reactors. This is called the airlift bioreactor. This draft tube basically creates a greater turbulence because the air bubbles are not able to escape and be evenly distributed. They have to move this through this draft tube. The airflow and air pressure creates a sort of a drag which creates greater turbulence, which ensures sometimes that there is greater mixing. Particularly when there is a high density culture, this type of draft tubes help to create greater drag, which conventional bubble column reactors don't necessarily create the same amount of turbulence. Now there are flow tubes. These can be parallel flow tubes or spiral flow tubes. Once again, these are designed to create greater drag and greater turbulence, which leads to greater mixing. Now, these are flat plane, unlike bubble column reactors, which are cylindrical, or air-lift bioreactors, which are cylindrical, or even these tubes, which have the same shape. Flat planes offer a greater surface area. However, they are much more difficult to operate by maintaining sterility and aseptic conditions. And this is, of course, a conventional bioreactor, which can be improvised to cultivate microalgae, which have impalers. So these are start tank reactors, but conventionally start tank reactors are made with some kind of metal or plastic casing. But if we are to cultivate microalgae, we must have some transparent plexiglass material, which would make them rather expensive. So all these systems, although highly efficient, much less susceptible to contamination, produces a greater drag and therefore greater biomass, they are rather expensive because we have to maintain the aseptic conditions, the sterile conditions, we have to maintain air pressure, and a lot of other factors. The final example, which I will give in the next slide, I will illustrate in greater detail. These are known as raceway ponds. These are open systems. So as you can see here, these are the name comes from the shape of the engineered system of microalgae cultivation. This looks like a raceway where cars can or horses can race in a circular loop. Here we have these impalers, which allows the mixing of the algal growth medium by producing a turbulence, and the algal mixture is constantly mixed within this raceway pond. I'll show a small video. So as you can see here, this is a 3,000 liter raceway pond where microalgae can be cultivated on a fairly large scale. And these impalers are causing the mixing and these impalers are causing the mixing, and slowly the microalgal cultures are getting mixed to ensure there is sufficient amount of aeration as well as a fairly homogeneous culture system. So moving on to some of my research work, I have worked with microalgae systems. I started working during my postdoctoral research at the laboratory of Professor Ramkrishna Sen. It was a project that was partly funded by the Department of Science and Technology, Government of India, and there was also Hindustan Petroleum, which was one of the implementing partners. So the work was done largely at the Indian Institute of Technology at Kharagpur. So I will just present briefly that work, which will give some practical perspective into where we stand in terms of research on this particular area and what are the challenges of microalgal cultivation that I presented right now, and what is the promise of potentially viable, sustainable solution. So we started with by designing a miniaturized thermal power plant. The Department of Mechanical Engineering was very kind to help us with this design and construction of this power plant. We obtained coal from Kolaghat thermal power plant, and we used this miniaturized system which generated flue gas. So this was an indigenously developed flue gas generator, and the operations were in almost entirely same as the one that operates at Kolakhat. So we have the stack, we have the cooling jacket, we have the coal furnace, we have the water pump, we have the compression systems to collect the carbon dioxide. So basically we were trying to study, because thermal power plants operate at a monumentally large scale, it becomes very difficult to study these systems in a comprehensive manner. So we designed and miniaturized the thermal power plant design, and we collected flue gas. And so this flue gas is essentially the same as what is used, or rather what is produced in a typical thermal power plant. So we characterized this particular flue gas that was coming out. As we can see that the carbon dioxide percent was fairly high. It's about 10 plus minus 2%. Oftentimes it would be, sometimes it would go up. Typically, with the kind of coal that is widely used, it can go up to 15%, particularly if the quality of the coal is not very high. So we had carbon monoxide, oxygen, oxides of sulfur, and oxides of nitrogen, and of course hydrocarbons. Now, most of these emission gases can be treated, and we have technologies to deal with them. And the vast majority of a significant component, that is fly ash, is used as a viable construction material. The only problem happens with this carbon dioxide gas, and we are emitting very large quantities of this into the atmosphere every year. Right now, the last time I checked the database, I think if you go to the website of NASA, it's more than 400 ppm. The last time I checked, it was more than 410 ppm. Obviously, the data changes and fluctuates on a regular basis. So let's assume it's around 410 ppm. So like I said, that carbon dioxide is a source of dissolved inorganic carbon. Is an example of inorganic carbon. And we have to ensure that microalgae, which are aquatic microorganisms, which largely live in aquatic environment, the carbon dioxide gas is made available to them. So it has to be presented in the form of dissolved inorganic carbon. Now it can exist in three forms. It can be in the form of carbonates, it can be in the form of bicarbonates, and it can be in the form of carbonic acids, which is rather sparingly soluble in water. And it dissociates into carbon dioxide and water. So carbon dioxide, sparging carbon dioxide, there has been extensive studies and attempts to sparge carbon dioxide directly into algae cultivation systems with very little success. Because of the low solubility, the vast amount of carbon dioxide that is being sparged either escapes or it causes a high degree of toxicity. The other two forms are, of course, like I said, carbonate and bicarbonate. Now, carbonates are usually soluble at a very high pH, pH greater than 12 or 10. Typically, that is not considered to be an ideal pH for microalgal growth and algal physiology. The bicarbonates, on the other hand, are soluble at a pH around 8, which is considered to be the ideal physiology, which is closest, I would say, to the physiological pH of microalgae. So we started our experiment by using sodium hydrogen carbonate, that is sodium bicarbonate. We can produce bicarbonates from many other ways, the easiest being making an alkaline solution and sparging carbon dioxide, which would lead to the production of bicarbonates. The rationale behind using sodium bicarbonate or sodium hydrogen carbonate is that it is also a very affordable source of inorganic carbon, dissolved inorganic carbon, because sodium hydrogen carbonate is essentially baking soda. So it's a very common household chemical, and therefore this can be a good, I would say, proxy or substitute for using carbon dioxide gas that is being sparged into an alkaline system. So we started by determining how much carbon dioxide can be tolerated by microalgae, or rather how much bicarbonate can be tolerated by microalgae. While working with microalgal systems, we used a model strain, that is Chlorella vulgaris. Now, just like in the study of yeast, we use Saccharomyces cerevisiae, or in the study of bacteria, we use E. coli. Chlorella vulgaris also happens to be a model strain of microalgae, which has been extensively studied and therefore an ideal candidate to start any kind of novel study in the domain of algal biotechnology. So we used various concentrations of microalgae, starting with 0 millimolar, then we use 50, 100, 150, and 200 and 250 millimolar. And the media that we used is Bold's basal medium. Any basal medium is typically devoid of any carbon source. So we supplemented the minimal medium that was devoid of any carbon source with differing concentrations of sodium bicarbonate in the concentrations that I have mentioned here. What did we observe? First and foremost, we observed that with increasing initial bicarbonate concentration, yes, the growth of microalgae does increase significantly, but 150 millimolar appears to be the threshold limit beyond which the microalgae is not able to tolerate the bicarbonate concentration. And so for further experiments, we used only these 4 concentrations, where 0 millimolar bicarbonate is essentially the BBM or the control medium. And we performed all the subsequent experiments with 50, 100, and 150 millimolar of bicarbonate, initial bicarbonate. So what did we observe? We observed that for BBM, the growth is staggeringly low. It's hardly around 0.75 to 1 gram per liter. And with 150 millimolar bicarbonate, it is almost 2 grams per liter. Interesting. The most interesting observation in this graph is, and I will come back to this later, or rather we will see why this is important. Let's take the 50 millimolar initial bicarbonate concentration. We see that initially, up to day 3, the growth is highest in case of 50 millimolar. But with higher concentrations, the growth increases with the progress of time. Why is that? We will investigate that later and we'll come to the explanation later. We observed other pigments like chlorophyll A, chlorophyll B, total carotenoids, and pH. And we found that, yes, they almost all of them follow the similar pattern, where 50 millimolar initial bicarbonate exhibits maximum growth and 150 millimolar initial bicarbonate concentration produces maximum growth after the completion of reactor operation from day 0 to day 7. Same with chlorophyll B, total carotenoids. And of course, the pH, like I said, because the microalgal growth turns medium alkaline, we were able to observe that, yes, when there is enhanced growth, the pH does indeed increase significantly. So what were the key takeaways from these experiments? The chlorella vulgaris exhibited maximum growth at initial concentration of 150 millimolar. The initial growth rate, however, was higher with 50 millimolar bicarbonate, and pigment synthesis also was higher with 50 millimolar bicarbonate in the initial phase of growth. Now, let's look at what happens to the residual bicarbonate, which means that when I am adding bicarbonate, how much of that bicarbonate is the microalgae able to utilize and how much remains unutilized at the end of reactor operation. Obviously, because there is no bicarbonate exogenously added to BBM, it's almost undetectable. And with 50 millimolar bicarbonate, it is also very low, which means that while we are providing 50 millimolar bicarbonate, the microalgae consumes the vast majority of it. But with 150 millimolar bicarbonate, some residual bicarbonate is indeed left behind, which is about, let's say, 25%. This is a very interesting observation. We find that how much carbon dioxide is fixed in terms of milligram of carbon dioxide fixed per liter per day, so per ppm per day. And we find that with 150 millimolar bicarbonate, the inorganic carbon that is fixed is close to or very in and around 400 parts per million. What happened? Is my screen visible? Is my screen still visible?
Yes, yes.
Okay, thank you. Okay, thank you very much. Another interesting observation we made, that once we completed the reactor operation, and if we leave the bioreactors undisturbed, gradually the microalgae tends to self-flocculate, which means the microalgae starts to settle down. And if you look at the flocculation efficiency, it seems to be proportional to the initial bicarbonate that we add. So if we add higher bicarbonate, the flocculation efficiency is higher. So the carbon dioxide consumption rate, this is the amount of carbon dioxide that is fixed by the microalgae, there is almost a 38% increase. And in terms of flocculation efficiency, there is almost 55% increase compared to the 0 millimolar. That is the control. So this is a picture. After we run the reactor for, let's say, 7 to 8 days, we find that the microalgae has settled down. So this raises a very interesting question. Why does microalgae self-flocculate after the reactor operation has been completed? So we looked into it and we tried to investigate why does it happen? And also there is the question of why is there indeed increase in the biomass production because of addition of the sodium bicarbonate at particularly 150 millimolar concentration? What happens? So we started our investigation by looking into one simple factor. Bioaggregates cause flocculation, or rather aggregate formation is one of the major reasons for self-flocculation. What causes aggregate formation? We know that extracellular polymeric substances, that is EPS, is one of the major constituents of flocculation of microalgae. But we found that there was no change because EPS is also associated with attachment of a microorganism to an abiotic surface. We found that there is no significant alteration in the attachment profile, or there was no discernible pattern at least. So perhaps this bioaggregate is formed of polysaccharides or other polymeric materials that helps the microalgae to form a robust bioaggregate, and perhaps that is the reason for which there is indeed enhanced growth as well as enhanced flocculation. So this estimation was followed by a simple epifluorescent microscopic analysis, and we found that yes, indeed, with increasing initial bicarbonate concentration, the microalgal cells seem to be associated with some kind of aggregate formation. So we went further and we tried some high resolution microscopic imaging, because this was a fluorescent microscopy which did not yield any significant result other than indicating that yes, there is something indeed going on which needs further investigations. So at this point, we call this biofilm or bioaggregate. One can debate whether we should call this a film or whether we should call this an aggregate, because there is no significant change in the attachment profile. However, with the subsequent analysis, particularly with high resolution microscopy, we were able to arrive at some, I would say, conclusion that what is the nature of this aggregate and what is the contribution, more importantly, what is the contribution of these aggregate structures. So first we did electron microscopy, that is SEM, and we see that, okay, this is pure BBM. There is no bicarbonate, so 0 millimolar bicarbonate, and the cells are quite discrete and distinct from each other. And there is no significant production of any matrix-like structure that is typically associated with EPS formation, aggregate formation, or even biofilm formation, because the most of the cells appear to be discrete and distinct. Quite clearly, if we add 50 millimolar bicarbonate, there is some matrix formation. There is some formation of aggregate-like structures, and the cells appear to be embedded in this matrix-like structure. If we increase the bicarbonate concentration, there is significant increase at 100 millimolar. The matrix becomes even more apparent. And by 150 millimolar bicarbonate, almost the cells are significantly embedded in this extracellular matrix. So if we compare this step by step, 0, 50, 100, 150, I think one can make this out fairly easily, that yes, indeed, there is this formation of aggregate-like structures. There is indeed this formation of matrix-like structures, and the cells are embedded within these matrices. So once again, same images demonstrates increased biofilm slash bioaggregate formation property with increasing bicarbonate concentration, and the algal cells are clearly embedded within the matrix. Then we went a step further. We performed another sort of high resolution microscopy, that is atomic force microscopy. Now, as we can see from this, that the cells, if we look at how atomic force microscopy works, there is a beam, a cantilever beam that scans the surface of the sample, and then a digital image is reconstructed from the data that is obtained from that scan. And clearly, here we have discrete cellular structures, very distinct and discrete cellular structures, and increasingly the cells appear to be more and more embedded. And if you look at the number of cells also, that is increasing, which is corroborated by the biomass data. So what do we find? We find that yes, with the increase of initial bicarbonate concentration, the AFM actually has further validated the biofilm slash bioaggregate formation property with increasing biomass concentrations. And the algal cells are once again, like I said before, clearly embedded within a matrix. Then from the AFM, the atomic force microscopy is not just about imaging. We get a lot of qualitative data as well, particularly cell size parameters, cell roughness parameters, surface to volume ratio, and these things. So if you look at the diameter of these cells are decreasing. Why is that? If we look at how an AFM, rather atomic force microscopy works, it essentially scans a three-dimensional surface. So if the cells are embedded within a matrix, the scan will reveal that the diameter has reduced. The same thing will be observed with topological height. But in terms of surface to volume ratio, we find that the surface to volume ratio increases with the increase of initial bicarbonate concentration. And the other other roughness parameters also indicate that with increasing bicarbonate concentration, there is some decrease in the roughness. So what does it mean? What does it mean when the cells are embedded within a matrix? What does it mean when the surface to volume ratio has increased? What is the physical implication, or what are the morphological implications? First, a smoother surface means there is easier exchange of gases and nutrients. An increased surface to volume ratio also indicates there is much more efficient and rapid mass transfer of nutrients. And so that is one of the reasons for which we find that the growth or the biomass production has increased, because the microalgae is able to participate in the gaseous exchange process, and the mass transfer of nutrients, the process of mass transfer of nutrients and the gaseous exchange, both critical factors for microalgal growth, has increased with the increasing bicarbonate concentration. And so we can conclude, or we can come to this conclusion as to why there is an increase in the biomass concentration. Furthermore, the, the matrix of this bioaggregate provides a stable microenvironment and a stable niche for this microalgae. In an otherwise hostile environment, there is turbulence, there is purging of air, there is some competition for nutrients, some competition for lights. So with the progress of time, the growth medium becomes increasingly hostile. But this bioaggregate formation provides a very stable microenvironment and provides a very stable niche to these microalgae, within which the cells are able to not only thrive, but also increase their productivity. Now, like I said before, that in the initial phase of growth, there is indeed greater biomass production with 50 ml bicarbonate concentration. So why is that? We looked at the residual bicarbonate. We measured the residual bicarbonate concentration on a daily basis from day 0 to day 7. So if we have 50 millimolar bicarbonate, we find that after day 3, almost the entire bicarbonate has been used up, which causes the staggering of growth after some time. So we devised a new strategy. This is what we call intermittent feeding system. So this is not continuous growth like a chemostat or a turbidostat, and this is not strictly speaking a batch culture. So we devised a new strategy of intermittent feeding. What did we do? We tracked the bicarbonate concentration, and every time it came down, we added some more. We added some more bicarbonate. Once again, it increased, and then it started to decrease. When it decreased again, we added some more. And finally, by day 7, we find that almost the entire bicarbonate has been utilized. And of course, the microalgal growth is very high. And with this, we were able to break the 3 gram per liter threshold. Now, this is considered to be one of the most significant barriers in algal biotechnology, that is the low yield of microalgal biomass. And we were able to break the 3 gram per liter threshold. And if you remember, if you use conventional minimal media, it's hardly 1 gram per liter. So that was a very significant step that we were able to achieve. And in terms of biomass production, that is milligram per liter per day of biomass, it is the highest in this novel intermittent feeding strategy that we had devised. So if we compare this to other studies, there are hardly, at that time, there were hardly any studies which were able to break the 3 gram per liter threshold, which we were able to achieve. Other studies had crossed 2.5 gram, 2.2 gram. Sometimes it's much less when we use, let's say, wastewater or seawater instead of using pure medium. So with our study, we were able to cross the 3.2 gram per liter. And even more interestingly, if we look at the carbon dioxide that is fixed, that is carbon capture capacity of this intermittent feeding strategy, it is 460 ppm per day. So that is much, much higher than the atmospheric carbon dioxide. The amount of carbon dioxide present in the atmosphere is about 410 ppm. Just please give me a moment. Okay, so with this new feeding strategy, we can definitely capture more carbon than there is in the atmosphere. So this is one of the most significant take-home messages of this study. So this is the schematic. I think we can all understand this. We grew the microalgae with different initial bicarbonate concentrations. Highest was in 150. We looked at the flocculation. This was once again highest in 150. This is the flocculation, and we did some high resolution imaging that revealed that alterations in the topological complexity within the bioaggregate matrix facilitate bulk transfer of nutrients as well as diffusion of gases in the presence of increasing initial bicarbonate concentration. And that is what caused the increased growth as well as increased flocculation. The advantage of self-flocculation is that when we use microalgae, see, microalgae, the biomass is where the technology starts. It's where the downstream processing starts. So if the microalgae is able to self-flocculate, then automatically the cost of harvesting comes down significantly. So if we can reduce the cost of downstream processing as well, the technology will become more and more viable. So like I said in the beginning, that climate change is an engineering problem. Engineering problems in the 21st century require 21st century solutions. So what are the future plans? Global optimization of key parameters. We can use different type of statistical tools or machine learning tools. I think the very first speaker today, Professor Partho Mukherjee, spoke extensively on the use of machine learning and AI. So we can look into that. We can design new strategies which will help us to capture carbon dioxide. And of course, we have to use the harvested biomass for bioenergy production. So we were able to publish this work in Journal of Cleaner Production 2 years ago. So this was the paper that we had published. Now, if there is time, I think we have time. Yes, yes. So I'll just show a small video of how this entire algal cultivation system can work from laboratory scale to, let's say, a pilot scale. I'll show a small video where we can grow from bench to pilot scale. Now, this is not an industrial scale, although some industries have and are adapting this. I'm happy to say that, like I said, that Hindustan Petroleum was one of the implementing partners of the project. And at the end of this work, the work was actually, in fact, transferred. The technology was transferred to Hindustan Petroleum, who have also started production using this particular technology. There are several other industries in the private sector as well, like Tata Chemicals, Reliance Life Science, Hindustan Petroleum is also there, who are trying to adopt this algal biotechnology to produce, not only produce bioenergy, but also mitigate the impacts of climate change through carbon capture. So I'll play a very small video and end with that. So as you can see, these are bubble column reactors. These are simple bubble column reactors. These have about 3-liter capacity. And we are growing, or rather we are cultivating this microalgae under laboratory conditions. These are the 0.22 micron air spargers, which prevent any kind of contamination of the growth medium through the air. So we have to pass sterile air, and these filters ensure that the air that is circulating is sterile in nature. So these are the 3-liter capacity benchtop bioreactors. Behind there is the light system. This is, of course, the raceway pond. So like I said, this is an example of how we can go to pilot scale. So this is a 300-liter raceway pond. So an industrial raceway pond would be much bigger in size, 2,000 liter to even 20,000 liter. So here we have one of the operators who is adding this microalgal culture that was grown in the laboratory. He's adding this into the raceway pond. As you can see, he's demonstrating how we can do this. And these impellers are moving, and so the water is being constantly circulated. Here we can see 2 pipes that are sparking some kind of gas. This is essentially, I will show it later. And this is essentially the carbon dioxide gas that is slowly being sparged into this growth medium. And he's slowly adding this. This is how we add the inoculum. Now, of course, as you can see, this is an open system and therefore extremely susceptible to contamination. But there is very little technological intervention or energy input. All we need is a very large tank like this and a baffle and an impeller that will constantly move this at a very slow pace. So we don't need any kind of significant energy cost or input or any kind of other interventions. So as you can see here, this is constantly circulating the air. The baffles are moving very slowly, but the turbulence, because of this design of the raceway pond, even this very slow movement of the baffles can produce significant turbulence. And this turbulence will cause adequate mixing. And so the microalgae will be constantly mixed, and all the microalgal cells will get adequate amount of air, adequate amount of sunlight exposure, which will ensure that there is high growth. So after a few days, we see that, yes, indeed, the growth has increased. So like I said, this is the CO2 cylinder. I think we can see the label, and this is the hydroponic pump. So it is mixing, because pure carbon dioxide microalgae will not be able to tolerate. So we have to mix a bit of carbon dioxide with pure air, and about 2.5% of carbon dioxide works fine. And as you can see that from where we started, it is much denser and deeper green in color, indicating that there has been huge amount of growth. So that brings us to the end of the video. Once I joined this university, the Neotia University, Professor Prashant Trivedi was extremely supportive and helped us establish this green biotechnology lab. There were 3 dissertation students who started working in this lab. All we had was a room and the sheer determination of Professor Trivedi and these 3 young lads. This is Sumit Kumar Mondol, this is Shiv Prasad Kaur, and this is Ayan Singh Roy. Ayan was the topper of our batch. He has recently got the Inspire Fellowship to pursue his PhD. He's doing very well for himself. Shiv Prasad Kaur and Sumit joined the PhD program of this university, fortunately under my supervision. And so, well, fortunate for me, and I'm not sure about them. And these are some of the photobioreactor systems that we operate in our lab. And we are continuing our work that I had done during my postdoctoral years. So these are the references and my acknowledgement. And thank you very much for your patient hearing. I always end with the words of the great Madame Curie, that nothing in life is to be feared, it is only to be understood. Now is the time to understand more so that we may fear less. So that's the end of my lecture. Thank you very much for your patient hearing. I'm open to questions. Otherwise, I think, yeah, Dr. Paul.
Thank you, Dr. Dashgupta, for such an informative video and also a very inspirational story of how we can go on with research if we just have the space and the mindset. So,
And we have Kushal Trivedi also.
definitely that is there. So, thank you for your very insightful lecture. Now, I look forward to any questions from the participants. Do you have any questions?
I suppose not. So I don't think we have any questions. So yeah.
Okay, fine. So this brings us to the end of today's session. Tomorrow we have another lined up sessions and the details will be shared in the group. So please join accordingly. Thank you, everyone.
Thank you very much. Thank you, Dr. Paul.